First of all, I'd like to thank Jim for this stunning meeting, and it's always a pleasure to come here, and I'm also very grateful and honoured indeed to be leading off this session that's going to celebrate Bertels, I guess your contributions in part to scientific contributions. And I'm going to focus, because I know something about Bertels' background and what he's contributed to the area, I'm going to focus on the repair and tie it into the evolution of Photosystem II. And as we've all met, or as we've met so far, we've focused on the oxygen evolution, and we know that, all of us know, that Photosystem II is prone to damage. And importantly, it can be repaired. And this has attracted my attention over the last 15 or so years, this repair process, as well as the assembly process that's involved in the repair. And the actual damage to Photosystem II, there are many plausible mechanisms, and they're defined as acceptor-side, donor side, and the manganese cluster. The acceptor-side and donor side, this is something that Bertel contributed to. He actually performed some seminal studies with collaborators on looking at the mechanisms of damage to Photosystem II. And what's really fascinating about this particular repair process is, firstly, it's just one subunit, the D1 subunit, that's mainly damaged. And then once there is a damaging event to Photosystem II and it's inactive, that complex has to be recognized as being damaged, and D1 has to be stripped out. And the other subunits, the undamaged ones, are recycled. So that's a very energy efficient way of maintaining activity. And I got into this area really through Bertels and Jim's interest and publicity of this area. And this is a very, very famous review that they published, Too Much of a Good Thing Like Can Be Bad for Photosynthesis, back in 1992. And I just returned back to Imperial College at that stage. And each one of them is a force of nature. So you can imagine when both of them got together in London, there was a lot of energy and a lot of ideas. And I was absorbing these ideas and thinking, gosh, can I do better than these guys? Oh, but a lot of beer, yes. And in some of these slides, I've put down the number of citations, because I know Bertel's very keen on keeping a tab occasionally of how many times his papers are being cited. So this is 1,021 as of the 15th of March. And so this particular review summarized some of the ideas at that time. Essentially, you've got Photosystem II over here. Apologies for the poor reproduction. You can have acceptor-side, donor-side damaging events that are due to either problems on the acceptor side of the complex. You get triplet chlorophyll generating singlet oxygen, or perhaps you have on the donor side, you have aberrant oxidation by by YZ, P680 plus events in the manganese cluster. Either way, you get damage to D1, and it's degraded in some way. Shown down here, you remove D1, and then you have to insert a fresh D1 and reassemble the complex. So this clearly was an important process and something that attracted me as I was a young and promising scientist at that time. Now I'm not even young or promising probably, but so I was really intrigued by this, and and another classic paper reviewed by Arrow, Bergen and Anderson from 1993. 2,210 citations, a classic in the field. And this gives us the framework for how we consider the Photosystem II repair process. It's called here the photoinhibition repair cycle, which is more rigorously correct. I call it the Photosystem II repair cycle. Essentially, you've got active Photosystem II, you try and down-regulate it in strong light, but ultimately it becomes damaged by acceptor or donor side mechanisms. At this stage, you've got damaged Photosystem II, and then somehow you have to recognize the damaged Photosystem II, degrade D1 through some proteolytic process, and then disassemble it, reassemble it, and so on. So there are distinct steps in this process, and I've been interested in the degradation of D1 side, as well as the insertion of fresh D1 over here. Now, why it's of tremendous interest is that if the repair process cannot keep up with the rate of damage, such as at high light, you get a net reduction in Photosystem II activity and net reduction of photosynthetic activity, which has an impact in the field. So over here we've got plants that don't grow so well at high radiance. And there is an example here of bleached coral where the symbiont can no longer survive under the particular conditions that it's been exposed to. Now, where I got into this area was when I was wanting to know more about the potential proteases involved in degrading damaged D1. And here are two papers that Bertel was involved with. The first one here with Lindahl was where he identified a possible role of an FDSH protease in degrading damaged D1. It's all through in vitro studies, purifying enzymes, adding them to damaged photosystem II and seeing if they can cleave the complex, or D1. And subsequently, a paper with Ivona Damska showing that a DEG-P protease could also play a role in this removal of D1. And the model they came up with at the time was that the DEG-P protease, we won't worry about the details, recognizes damaged D1 in white, cleaves it, and you get fragments, and then the FDSH protease moves in and removes these fragments. So this was the starting point for the work we did about 15 years ago. We wanted to look for in vivo evidence that these proteases did have a role in photosystem II repair in vivo. And we used Cyanobacterium silicicystis, and we screened various mutants for effects on photosystem II repair. And what I want to discuss in this presentation is what we think the role is of the FDSH protease in D1 degradation, how is damaged D1 recognized, and how the evolution of photosystem II, the structural evolution of photosystem II, has been constrained by this need to replace damaged D1, because we feel there's an impact of that process on the evolution of photosystem II. Now, we identified a particular mutant, an FDSH mutant, that was impaired in photosystem II repair. There are 4 distinct protease genes, or genes encoding FDSH proteases. We identified one mutant that's affected in photosystem II repair and D1 degradation, and we isolated that particular form of FDSH by GST tagging. We purified it, and we saw that it was present as a complex with a second type of FDSH protease subunit. So it's FDSH2, FDSH3, and we did some rather crude structural analysis by single particle electron microscopy techniques to show that this complex was hexameric, it was a heterohexamer, and we had alternating subunits of these two types of FDSH in the complex. And so what does an FDSH complex do? Well, it's found throughout nature, and it has both it's composed of an AAA plus domain and a peptidase domain, shown here. It's a member of this type of protease. And this class of protease will recognize a target protein and use couple ATP hydrolysis with the unraveling of the protein into the peptidase, a protease domain for cleavage. So it's a highly processive reaction, and usually at the end of a target protein. So it could be the N-terminus or the C-terminus. And there's a little hydrophobic pore here through which the protein goes. And the mechanism of this type of enzyme complex is still not known, and something that we should really work on closely in the future. In fact, there's no structure so far for the intact holoenzyme. And so what we asked first, one of the early questions was, what is the nature of the degron, in this case of D1, that's recognized by FDSH to cleave it? And we thought that it's perhaps the N-terminal tail that is accessible and is not poorly resolved in the structures, but this N-terminal tail could be the degron that guides it into the FDSH complex. And to address whether that was feasible, we performed a very straightforward experiment. This was in collaboration with Josef Komenda in the Czech Republic, in which we just trimmed the N-terminal tail by 20 residues. So we shortened it by 20 residues, thereby preventing it with interacting with the FDSH complex, and thereby preventing efficient proteolysis. And this is the type of experiment we did. It's a pulse chase experiment. Here's the wild type. Under normal conditions, illumination conditions, D1 is rapidly labeled because it's turning over rapidly because of damage and resynthesis. And then you can see that you can determine the half-life of the D1 through this chase with non-radioactive methionine. And in our truncated mutant, you can see, first of all, there's less radiolabel and much slower kinetics of D1 degradation. And here's a repair assay, a classical repair assay, where we measure photosystem II activity as oxygen evolution, either in the absence of a protein synthesis inhibitor or the presence, and this gives you information on how effective the repair cycle is. So in the wild type, very effective repair because we can maintain photosystem II activity as a function of illumination time at high light, whereas in our mutant, we can't repair photosystem II because we can't degrade the damaged photosystem II. So this provides evidence that it is a processive process, that the FDSH complex recognizes the N-terminus of damaged D1, and then it attaches and degrades. So this is the best evidence to date about the mechanism by which D1 is recognized. So somehow in the damaging event, the N-terminal tail becomes accessible for the FDSH complex. So this is our current view of how damaged D1 is removed from complex, and it holds both for cyanobacteria and in chloroplasts. So here we have our photosystem II complex, D1, D2 at the heart, CP43, CP47 either side, and for the FDSH hexameric complex to degrade D1 by attaching to the N-terminal tail, we envisage you need to remove or detach CP43 to gain access because D1's right at the heart of the complex. So you need to open up the complex some way. So there's a detachment. We have a processive removal of damaged D1, and then the new D1, in the case of cyanobacteria, is provided post-translationally. It's not a co-translational insertion. It's there in the membrane, stabilized, then adds to the remaining subunits, and then you have reassembly and reassembly of the manganese cluster. So that's our model for D1 degradation. And this is the FDSH pathway. This is the dominant pathway. But there are other backup pathways when FDSH activity can't cope with the rate of damage. So if we go back to Bertels' early work, he suggested there was a two-step mechanism. You have primary cleavage, then FDSH-mediated removal of fragments. And our work suggested that FDSH was important for the removal of intact D1, damaged D1, not just fragments. And so we recognize now that the way that how damaged photosystem II is repaired depends on the degree of damage to photosystem II and the metabolic activity and how well the cells are to be able to have enough ATP to remove the damaged D1. So the first line of defense, when you damage D1 here and it goes from yellow to pink, this first line of defense, where we remove damaged D1, this is an FDSH-mediated process. And this will occur at low light and under optimal growth conditions. But under conditions where you can't remove damaged D1 promptly, you then end up with subsequent damage to other proteins in the complex, and then you have reactive oxygen species mediated cleavage, and you have a whole host of mechanisms whereby other proteases come in with lower efficiency to remove and repair the complex. So what I want to do now is just address the issue of the detachment of CP43 and how important that is for the repair process. And so the idea is how does the FDSH complex recognize damaged D1? Well, I've just gave you some data to suggest that it's the accessibility to the N-terminal tail is important. But in addition, we feel it's the detachment of CP43, maybe not far away, but a little way, to allow access and entry of the complex to damaged D1. And then we need to consider the similarities between photosystem II and photosystem I at this particular point, because it's been recognized, and here's a paper by Jim some years ago, 2000, there's a strong similarity in structure between photosystem II and photosystem I when you consider the number of transmembrane helices. And so in the case of photosystem I, there are two large reaction center complexes, PSA-A, PSA-B. Photosystem II, we've got D2 and D1, and they're split. So we have CP43, CP47 split. They're no longer fused with D2 and D1. So we have two subunits here, four subunits here. And you can see from the recent structural work that this similarity holds in terms of the structure. So it's likely that photosystem I and photosystem II are descended from a common ancestor. And during evolution, there's been selectional pressure either to fuse, to maintain these two large reaction center subunits in photosystem I, which is a type 1 reaction center with iron-sulfur electron acceptors. But in the case of system II, there's been a drive to maintain them as four separate subunits, to keep CP47 and 43 detached from the D1, D2 reaction center subunits. And of course, given what I've told you, we hypothesized that this split reaction center we see in photosystem 2 is really a evolutionary adaptation to allow efficient repair, because it allows CP43 to detach and have access of FDSH to the complex. So that's the question. Is it, this split reaction center, is it related to function, or is it related more to the repair process and to have prompt repair and removal of damaged D1? And so Shengxi Xiaowen, who's just finished his PhD, set off to try and turn photosystem 2 into a more photosystem 1-like reaction center by fusing CP43 and 47 to D1 and D2. So here's the structure of photosystem 2 from cyanobacteria. And over here, in blue is CP43, green D1. And you can see here's the N terminus of D1, the C terminus of CP43. And what he did was he fused them. He made a new gene construct where he fused the C terminus of CP43, the N terminus of D1, had a little bit of a linker. So now we're making a cyanobacterial strain where we're artificially generating a single large reaction center subunit. And we're going to look at what the effect is on function and also on repair. And the way he did it was he returned this new fusion where you've got a CP43 D1 fusion, PSBC, PSBA2 fusion, into the same locus. So you have PSBD, PSBC, that's the original locus. We just add in PSBA2, and it goes into a D1 deletion strain. So the only D1 being made is part of this fusion. And the PCR analysis confirmed that it was correct. And when we did some blots, we can see straight away, here's our fusion strain. We're doing a D1 blot. So straight away in the wild type, this is the expected size for D1. In the fusion now it's a lot bigger because now it's fused to CP43, so we've got CP43 D1 fusion. And with our antibody we use over here for CP43, it doesn't recognize our fusion because the epitope is actually at the C terminus of the CP43. We isolated some his-tag preps from the fusion and the wild type. And you can see straight away, this is what you would expect for a wild-type Synechocistus photosystem 2 his-tag prep. We've got D1, D2, PSBO, CP43, CP47, some FDSH contaminants, some other extrinsics. And in our fusion, we lacked D1 as anticipated. We lacked CP43. Instead, we have a new protein up here. This is the CP43 D1 fusion. And when we do some native gels, just to look at whether it forms complexes, what type of complex? It forms monomeric, dimeric complexes, just as in the wild-type sample. So this fusion hasn't prevented assembly, normal assembly. The levels of the complex are similar to that of the wild-type, just a little less. And importantly, in our complexes, there's no hint of degradation of the CP43 D1 into the CP43 and D1 form. So it's still maintained as a fusion. And when we look at the functionality of this reaction center, it produces oxygen, and the level of oxygen from whole cells is consistent with the differences in reaction center. And when you look at electron transfer, either on the acceptor side, QA to QB, or charge recombination, very similar to wild type. In fact, there's no hint here in the charge recombination of a portion of a subpopulation of centers that haven't assembled the cluster. So it's all looking very nice indeed. And of course, what's the effect on repair and photoinhibition? Well, we were very pleased to see that this fusion strain could grow photoautotrophically. And, oh, your battery's running low. Hey, mine is as well. So there's under lower to moderate light conditions, it grows fine. But under high light, where it's stressed, where it's being damaged, you cannot now grow. It cannot grow photoautotrophically, unlike the wild type. So clearly there's an effect in being able to counteract the damage during exposure to high light. And just to go through briefly the activity the activity measurements. Here's over here the repair assay. Wild type, you can have good repair. The mutant lacks the protease for D1 degradation, poor repair. And our fusion strain, very poor repair. The repair cycle is very, very, very much impaired. And when you look at the loss of the rate of D1 degradation down here, in the wild type, you get very, very good degradation of D1 when you add an inhibitor of protein synthesis. So you go down to 6% of the original activity in 4 hours. whereas in our fusion it's down to 50%. So there's an impairment in the ability to remove that fusion protein, and we suggest that's because you can't detach the CP43 away from the fusion. Has to be degraded in one subunit, and that's much more difficult for the strain, for the cyanobacterium to do. So that's evidence that there is a drive, a selectional pressure to maintain CP43 as a distinct subunit in photosystem II to allow prompt repair. Nothing to do with the function, but to do with the repair process. And so the question is, what happens when you remove CP43? And so we can generate strains where we remove CP43, so D1 is now always accessible. There's no CP43 to block. And in such a strain, when you remove CP43, you can't assemble the manganese cluster. It's very sensitive to donor side photoinhibition. And what you see is a very rapid D1 turnover in the light. But what was very surprising to us, that this rapid turnover is also observed in the dark, even when there's no damage to photosystem II. So our interpretation of this is that CP43 is actually stabilizing or shielding D1 from degradation under all conditions, and that D1 itself is very prone to degradation. So the trigger for D1 degradation is merely the detachment of CP43 to allow the FDSH complex to attack D1. How are we doing? So our model is that here is our native complex where CP43 is protecting D1. If you remove the CP43, your D1 can now be accessed by FDSH complex. It can attack the N-terminal tail and remove it even in the dark when it's not damaged. Under normal conditions, this complex will become damaged. There'll be some triggering event whereby CP43 is detached slightly from the complex to allow the FDSH complex access to the N-terminal tail to drive degradation. And at the moment, we don't know what this triggering event is, but it could be related to events around the manganese cluster where the CP43 subunit provides a ligand. So more recently, we've gone to the full hog and tried to make a photosystem I like photosystem II. So we've fused both the original fusion I've described, which is CP43 to D1, and also we tried to fuse CP47 to D2. So now we're generating a 2-subunit photosystem II reaction center. And again, the ends are very close together, and we could just bridge the ends by providing a linker. And we generated, it's a rather complicated experiment because you have to generate a strain where you delete all the photosystem II genes and then reintroduce an operon where we've got all 4 of those subunits, genes encoding those 4 subunits in one operon. And we've only just, a final year undergraduate project students, been studying this strain. And nicely, we see that in this, we call it the big fusion or the unsplit reaction center, it definitely does assemble. So we have, here's our wild type, these are blocks, D1, CP43, D2, CP47. D1 is present as the largest fusion, CP43, the same fusion. There's a bit of degradation. D2, large fusion, CP47, large fusion. So this is good evidence that we're forming a 2-subunit photosystem II reaction center. And we haven't done a complete analysis yet, but it's quite clear. Well, it's not that clear here, actually, because it's faded away, but I assure you there is some oxygen evolution. So this reaction center is able to split water. And it doesn't grow photoautotrophically, so it's very, very severely affected with its ability to withstand high light. Okay. So just to summarize what I've told you, following on from the work that Bertel initiated, we followed that up and looked at the role of FDSH proteases in vivo, and it's now clear there are a family of FDSH proteases in both in cyanobacteria and in chloroplasts, but there's a subset of heterooligomeric FDSH proteases that are involved in removing damaged D1. They have other housekeeping duties as well, but they have a role in removing damaged D1. It's likely that the main pathway is a N-terminal processive reaction, and that from our experiments where we fuse CP47 and 43 to the reaction center subunits to generate single subunits, we argue that the split reaction center seen in Photosystem II, extant Photosystem II, is an adaptation that promotes efficient repair and is not related to any functional aspect. And that detachment of CP43 triggers D1 for selective degradation even in the dark. Right. So that's sort of what I'd like to tell you about the research I've been doing as a young to less young promising man. And I'd just like to finish off with this, to discuss or highlight some recent events in which Bertel has had an impact on my life. And I'm sorry, let me just thank the guys involved, Jianfeng, Shengxi, Anna, who's a final year undergraduate project, James Murray, who's involved with this project, and my collaborators, Joseph and Roman. They've all been involved in this work with the fusion. Some time ago, probably 2 or 3 years ago, Bertel came to my office and made me an offer I couldn't refuse, and he invited me to come to NTU as a visiting professor to join Birgitte's group to try and develop synthetic biology in cyanobacteria. So here's the group here. There's Tiago, Lifang, Artur, and Jasmine. And they're working very hard here at NTU to try and it's called Cyanosymbio at NTU, to try and manipulate or develop tools to generate new novel cyanobacterial strains to produce various types of molecule. And we're looking at lactate, hyaluronic acid, free fatty acids, and other value-added compounds. I'm not going to go into the details, but we felt it was very important at this stage that we isolate a Singaporean cyanobacterial strain that was fast-growing, that could be a useful chassis, a useful host for our experiments. So we went, I didn't, but Tiago and Artur went out to the Straits over here, and they isolated from a fish farm a particular strain, cyanobacterial strain, SSW07, a Synechococcus strain. And it has, and I'm sure Bertel will recognize many of the characteristics of this strain that make it a very good organism to survive in Singapore. So it's like Bertel, it's very active and efficient. So it grows as fast as the current record holder. It's got a doubling time of 2 to 3 hours. Very robust. This is so important if you're living and working in Singapore. You've got to be able to survive the high temperature, the high light. I'm not so sure about the salt. You may not need to worry about that, but you've got to be robust. Adaptable. This organism is very easily transformed. So it's adaptable. It can be transformed, and I'm sure Bertel had to adapt a lot when he came to Singapore. And it's very flexible over here. I don't know how this organism can grow as single cells, but also can form little filaments. So I'm sure Bertel's single-minded. He can work by himself, but also work in teams and so on. So that's where I'm going to finish. Thank you for your attention, and thank you, Bertel, for inviting me here as a visiting professor. Thank you.
Thank you, Peter. Thank you for keeping time and for humoring Bertil, which I think is very good. There is time for 3 or 4 questions. Bertil has the first one.
It was a very elegant continuation. I guess, of course, if I stayed in the field, I have done it myself much quicker.
Definitely.
Anyway, I thought it was great. But when you took away CP43, sort of in the dark, and you get D1 to degrade, I think one has to be cautious, because if you muck around with the complex and destabilize it, you have cleaning up proteolysis rather than the specific one. So there in the dark experiment, when you take in CP4, well, the other PS2 subunits were degraded as well.
Oh yes, I didn't. There is a hierarchy, but it's D1 is specifically degraded, and the important point is that CP47 and 43 are resistant to proteolysis. It's D1 and D2 we think are pre-programmed to, as long as they get into the complex, they're stable. But the minute the complex disassembles to some degree, they're immediately removed. So that's the way it's controlled. It's the disassembly that actually triggers the automatic removal. And of course, having a split reaction center makes complete sense, because you only want to replace the specific part of your reaction center that's damaged. And Jim and you highlighted many times the fact that D1 is specifically damaged because it's the active arm of the reaction center. It binds the manganese cluster, it binds the chlorophylls involved in charge separation, and so on. So it's where the singlet oxygen is.
It's also an evolution evolutionary pressure. It's a lot of energy required to
Yeah.
drive the repair cycle. So it's not trivial for the organism actually.
No, that's true. So, yeah.
Any more questions? Yes?
Yeah, Peter, it's really interesting. But the model then is that degradation or some inhibition of D1 then triggers a weaker binding of CP43, and you suggested it might be the manganese cluster. Does this have some implications on donor side versus acceptor side photoinhibition and repair? Is there some way that can be fit into this model?
Well, I have to say I've always been a favoured donor side photoinhibition, I have to say, and so I believe that it could be some effect around that part of the complex. And of course, we can go in now. There are plenty of mutants, the CP43 ligand mutants, the GLU354. They may be like the mutants I've described here. They may turn over D1 in the dark because they're more easily detached because they can't bind the cluster. Yeah, so it can be tested. James. Oh, sorry.
Yes? You start.
Thank you. That was very enjoyable, Peter. So, following on what Gary just said, what do you think it is which triggers which makes the CP43 come off?
We honestly, we don't know. We don't know. There are many possibilities. One of the interesting things is that where the C terminus of CP43 and the N terminus of D1 come together, the CP43 subunit has a little hook that goes over the N-terminal tail of D1, and it just goes over the end. So it's as if once you remove that, the N-terminal tail comes up. So it could be a very subtle detachment, not a full-blown detachment, a very subtle detachment, and it could be long-range effects that just manipulate that particular region. But obviously there's damage to pigment, there could be lipid peroxidation. There could be a whole host of effects that could contribute to this detachment. But I don't want to give the impression it's a full-blown detachment. It just may be a loosening of the structure at that site. Jim. Oh, sorry.
I think it seems a very good idea that the movement of CP43 is a trigger. But what about triggering the ribosome to synthesize a new D1 protein? Any idea about that? Maybe a fragment.
Let's see. For cyanobacteria, there's a pool of pre-made D1, and sitting in the membrane that can go in. But I don't know what the mechanism would be to fill up that pool. That's unknown.
Two last questions.
What is the action spectrum of photoinhibition?
Where am I looking? Oh, sorry. Oh, there.
Here.
It follows chlorophyll, and there's a bit more in the blue as well. So there's discussion about
Blue would be manganese.
Yes, that's right. So there's a bit of. It depends on who's done the experiments.
What is stronger? What is stronger? In normal life.
In normal life.
What is?
Oh gosh, I haven't done those measurements, so
Okay,
I can't say. It's a very. Imre Vass can talk about that because he's done those sort of measurements.
So actually, your system now, I think maybe it's an interesting. Actually, now you make the PS2 like PS1. So while you compare the actually it's not PS2 at all. Now it's close to the PS1. Maybe it will be nice to compare with the PS1, the function of the PS1.
Well, it's not got iron sulfur clusters and so forth. It's a photosystem II that's just now is formed as two big subunits. I mean, it's amazing, really, because we're asking the cell to assemble something it's never seen before, and to coordinate things, subunits, into a complex that it's never seen before, and it's managed to do it, and it produces high levels of the complex. So it just emphasizes how robust the whole process is. It's amazing.
So let's thank Peter once more.
Thank you.